Okay, so one of the starting points of my presentation since I turned 50 is this figure here, which depicts the enormous increase in lifespan that humanity has gained over the last few centuries. I mean, if you look at simply at the beginning of the 1900s, life expectancy was less than 50 years. At the beginning of the 2000, it rose to 76, and these are the last statistics from the WHO, and which say that in country like ours, Italy has a life expectancy of more than 80 for men and more than 85 for women. The top scorer are Iceland and Switzerland for men, but you see that the first top 10, I mean, there is not much difference, while for women, Japan is almost 87. And the reason I'm showing this, it is because, I mean, one thing that I find among the most fascinating is trying to understand what is the limit, and the limit statistically and epidemiologically will not be longer than 125 years for men, for the human species. And what I find really fascinating is trying to understand why, because there is a defined lifespan for all species living on planet Earth. So basically a rat lives 3 years, a mouse 2, dogs, that's a common experience, 15, 30 years, Drosophila 3 months, and so on. So there is a sort of clock embedded somewhere, evidently somehow written through a genetic program in our genes that sets the maximum lifespan for each species, and nobody knows where it is. So there is really no clue why a mouse should live 2 years and humans should live 120 years. There are many reasons why this happens, actually, which have to do with the fact that evolution is driven by genes, and what genes are transferred to the progeny. This is a driving force. So having a progeny that is self-sufficient to live on the planet and reaching self-sufficiency is much longer for a man than for a mouse. But where the clock is in molecular terms is completely unknown. What we start knowing, however, is that this progression toward this limit of 120 is somehow accompanied, or can even be caused, by the progressive exhaustion of the regenerative capacity of most organs, which happens after birth. So before birth, basically all organs, including the brain and the heart, are highly regenerative organs, and after birth, all organs in our body, either soon after birth or later, lose the capacity of regeneration. This is dramatic for the heart and for the brain. We are born with about 100 billion neurons, and we lose 80,000 neurons every day here, so we are losing a lot of neurons while during your visit here, and same for us, which is really scary. If you make some calculations, it turns out that a person after 8 years old has lost up to 10% of his brains or her brain cells, which accounts for all the symptoms or neurological degeneration that accompany age. Same was true for the heart. For example, I mean, from an adult heart, you can recover cardiomyocytes, which are these beautifully brick-shaped cells, and you can put them in culture. You can do all your electrophysiology, you can do all the molecular studies, you can do imaging, you can see them contracting. They are perfectly functional cells, but there is no way that these cells proliferate either in the laboratory or in vivo. But even the hematopoietic system, there was a fantastic paper last year on a lady who died in the Netherlands at 113. And this lady was followed. She died because of cancer, actually. But she lady was followed in the last 10 years of her life, and there were blood samples taken. And people asked them how many hematopoietic stem cells were sustaining her hematopoiesis. And normally, hematopoiesis from a normal human being is sustained by about 10,000 cells, which fire at different times and give rise to all the progeny, so red blood cells, white cells, and platelets. Well, in this lady, 113, the whole hematopoiesis, which was fully functional, was sustained by 2 clones. So she had exhausted the 10,000 cells down to 10 clones, and so it was good to have been expected that if time passed, these 2 clones became 1, and then she would die because of aplasia instead of cancer. And this is, in my view, what happens for most organs during life. And so it is not surprising that as we increase our life expectancy, also we increase the rate by which we suffer of the incapacity of the heart to pump, so heart failure, the generation of chondrocytes or arthrosis. We don't have any capacity to regenerate beta cells after they are gone, and we don't have regeneration in the brain, so we have dementia and all the age-related characteristics. I always tell also this, I have a friend in the United States, a collaborator who started a seminar looking at people in the audience and asking people, each one of you now should look at a person on your left side and the person on your right side, and then you have to decide which of you 3 will die because of a cardiovascular disorder. So the reactions are very emotional, so not only the same type of emotional reaction we could have here in Italy. But this is a reality, because 1 person out of 3 in the world dies because of myocardial infarction or stroke, 17 million people worldwide. And once the heart is damaged, there is no formation of new myocardium, and basically eventually this in most people configures a situation of heart failure, and after heart failure is diagnosed, 15 million people every year diagnosed with this condition, the probability of not being alive is more than 50% only after 4 years, which is a prognosis that is much worse than several cancers. If you would think that this is a problem of our societies, which are well developed, you would be wrong, because again, according to the WHO, the 80% of cardiovascular disorders are in low and middle income countries, in the big metropolitan cities of Asia or of Africa. People die more of cardiovascular disorder, metabolic syndrome, diabetes, degenerative condition than they died of all causes of viral infectious diseases only 20 years ago. You could say there are drugs against these conditions, but look at, for example, for the most prevalent condition, that is heart failure, the drugs that are available, and along with the timeline when they have been developed. So there are 6 classes of drugs that clinical cardiologists have in their armamentarium for therapy of these patients. Digitalis and diuretics are long known in the medical literature, where actually digitalis was used also anecdotically by the old Egyptians. And then in the 60s, the spironolactone, so aldosterone antagonists, the 2 drugs that changed really the fate of heart failure in terms of treatment, the ACE inhibitors, the beta blockers, were developed in the 70s. And in the 90s, the sartans, so the angiotensin II receptor blockers, which means that a person who is admitted to an hospital here in Trieste, but also in Riyadh or at the Mayo Clinic or at McGill University Hospital now, he is actually treated for heart failure with the same classes of drug he would have been treated 25 years ago. So it's 25 years that neither the pharmaceutical industry nor the academy have been able to develop new compounds. And this is true for heart failure, but this is true for Alzheimer and other dementias. For example, in over 80 years in Europe, 30% of people, according to the European Union statistics, develop a dementia, so 1 person out of 3 again. And there is no treatment at all against dementia. There is no treatment for the more than 170 million people with diabetes to promote the regeneration of their beta cells that are gone either through an autoimmune attack in type 1 or through exhaustion in type 2. We had a very interesting conversation yesterday at dinner about malignant diabetes in children now in Saudi Arabia, by which these children, by the age of 18, they exhaust completely their beta cells and they become insulin dependent for the rest of their life, which is definitely shortened. After 75 years, 1 person out of 3 don't see well, and one person out of 2 doesn't hear well because of degeneration of the retina and degeneration of the neuroepithelial cells of the inner ear. We be not hearing well during aging is something that we consider as a common thing associated with aging, but indeed it is a disease due to the loss of cells. So this is the reason why the main theme of my laboratory and of several others is to try to find ways to develop new biotherapeutics for these conditions. And these new biotherapeutics should be biological agents, so could be genes, for example, but not only genes in the sense of coding DNA, but these genes could be any kind of nucleic acid, so a small regulatory RNA or an anti-RNA, which can be delivered in various means to the bodies, or could be synthetic peptides or recombinant proteins, or obviously could be cells that regenerate or that somehow supply functions that have been lost during aging. So this is our broad aim, where we want to achieve. The good news in this, at least experimentally, is that there is a class of vectors, also modified viruses, that work extremely well exactly in the same cell populations that undergo degeneration. This class of vectors is based on the virus, which is called the adeno-associated virus, a very unlucky name. It was discovered 40 years ago as a contaminant of cultures infected by adenovirus, but it has nothing to do with adenovirus. It is a small virus belonging to the parvovirus family, single-stranded DNA molecule with 2 herpes at the 2 extremities, very small, perfect nano-sized particle, 20 nanometers in diameter, 1 single RNA surrounded by 60 proteins. Very, very simple. Genetically, it's also very simple. It has only 2 genes, rep and cap. You can remove completely these 2 genes and substitute with the gene you want to deliver inside the cells. You can produce these genes very, very simply. So basically, you clone a plasmid with this kind of construct I just described you, and then you have a reciprocal plasmid with the 2 AAV genes, rep and cap, plus some genes from adenovirus that help replication. You transfect these simply with calcium phosphate in cultured cells. The cells, after 48 hours, produce 10 to the 4, 10 to the 5 particles per cells. They are completely lysed by viral production. You can recover the virus by simply biochemical means. You can use this virus for transduction. Now, if you use this virus for gene transfer into any culture cell in the laboratory, so all tumor cell, but also primary cells that replicate, like endothelial cells, hematopoietic stem cells, keratinocytes, fibroblasts, and whatever cell you like that grows in culture, they are very, very bad. So they are the worst vector you would use. Never use an AAV vector for gene transfer in cell culture. However, if you take exactly the same preparations, you inject them in vivo, these vectors are fantastic. And they go to tissues like skeletal muscle fibers in the muscle, but not fibroblasts, for example, not endothelial cells. Cardiomyocytes in the heart, but not fibroblasts or endothelial cells. In the brain, to neurons, much less to glia. In the retina, to all cells in the retina. They go to beta cells. There are some variation of these 60 proteins that form the capsid we call serotype that allow this vector to be injected systemically, and they go to muscle and heart. And the efficiency is really very, very high. So you can transduce more than 80%, from 80 to 100% of cardiomyocytes in the heart if you inject in the neonate, and you see that they really go to the cardiomyocytes. And what have all these cells have in common? So skeletal muscle fiber, neurons, cardiomyocytes, beta cells, or retinal cells, is that they are post-mitotic. So they belong to the category of cells that exit from the cell cycle and will never re-enter the cell cycle. An endothelial cell is also a resting cell, but every 6 months all our endothelium changes, while a neuron is a neuron with which one is born and will never change all through his life, or a cardiomyocyte is exactly the same, a retinal cell is the same. And so they have this peculiar tropism. We were very much intrigued in trying to understand why this tropism is there. So here in Trieste, we have a facility for high throughput screening, and I will detail on this tomorrow. Unfortunately, Miguel Mano, who is the coordinator of this facility, had to return to Portugal, his homeland, and so I mean he left me his presentation. I will show you just some details on this of the work we do, but basically we have a facility for high throughput screening and we have libraries corresponding to siRNAs against all the human genes and all the whole genome microRNAs of human, mouse, and rat origin, and the inhibitors for these microRNA. And so basically we did a high throughput screening asking which are, out of the 20,000 genes of the human genomes, those that determine permissivity to AV transduction. We run the screen. All the genes that are around 0 are genes that are irrelevant for AV transduction. But we found that there were, out of 20,000, almost 1,000 genes that significantly increase AV transduction. Some of these increase even 50-fold the capacity. And the screening was performed in HeLa cells, which are not very permissive. And we were surprised to learn that among the most effective knockouts were knockouts for proteins that belong to the DNA damage response machinery. So the genes that control genomic integrity in the cells. So the genes that recognize damaged DNA and trigger the DNA damage response. And basically, to make a long story short, what we believe is happening is that in a cell that is non-permissive for transduction, like a cancer cell or any culture cell types, the virus enters these cells. It is recognized as damaged DNA because it has a single-stranded DNA, has these two hairpins that mimic DNA damage. And these cellular proteins recognize this as DNA damage and block conversion from single strand to double strand, and basically they block the capacity of the virus to transduce. And just one image that I like it very much to corroborate this notion. This is another experiment that we did by constructing AV vectors with 112 repeats of the LAC operator, so a very simple genetic sequence, and then produced a virus. And the virus was used to transduce cell that in the nucleus expressed the LAC repressor fused to GFP. So basically, the nuclei of these cells are uniformly green. And then one of the virus comes in, and this is a poorly permissive fibroblast cell line. You see that the virus starts accumulating these dots inside the nucleus, which was very surprising to us. You see that these dots grow larger in size at different times after infection. And they are pretty stable. They remain there. And we spend a large amount of time in trying to understand what these dots are. And eventually what we discovered was that these are exactly the same places where proteins of the DNA damage machinery also go. For example, you can co-localize these AEV foci with foci where MBS1 or MR11, RAD50, MDC1 also localize. So these are proteins that are activated in the cells when DNA damage is activated. And so that's a strong point towards the fact that these are the proteins that are inhibitory to transduction. You would say, but then why a neuron or a cardiomyocyte is permissive? Simply because they don't have these proteins. If you test the levels of these proteins in the heart and the skeletal muscle, it is extremely low compared to, for example, high replicating tissues like a bone marrow and spleen, which are not permissive. And the heart is a highly replicating organ immediately until birth, and immediately after birth it stops replicating. Serena will tell you more about her research in this respect. You see that these are days after birth. So if you look at a mouse heart at 0, 3, or 7 days after birth, there is a high level of expression of this protein, and then these proteins are completely switched off. And since these cells don't replicate, they will remain off for the rest of the life. In fact, if you take an AV vector, you inject immediately after birth, you see some cardiac transduction. But if you wait 7 days or 21 days, this transduction is really massive. And this also has some biotechnological implications, because you can inject an AV vector into the liver, which is not so well transduced by the vector. You see some green cell. This is an AV vector expressed in GFP. But if you co-inject a siRNase against some of these proteins, MR11, RAD50, and MBS1, you see that the efficiency of transduction is extremely increased. So whatever the mechanism, we are very fond of these vectors, and we have a facility here in Trieste for the production of these vectors. There are technicians under the coordination of one of our staff scientists who produced these vectors, and basically this works all through the year. We produce every week at least 4 viral preparations. And so we have summed up in the last 10 years thousands and thousands of preparations. We collaborate virtually with laboratories all over the world on these vectors in the United States, Canada, Australia, Korea, Japan, and obviously Europe. And let me skip this for the sake of time. And we have been using this vector a lot as a sort of molecular tools for gene investigation. If you have your favorite gene and you want to know what your favorite gene does in one of these tissues in vivo, you can do a transgenic animal in which you switch on your gene by the tamoxifen increase system in the adult life, and this will take you 1 year of work if you are good. With the AV vector technology, you can simply clone a vector. In 15 days, you have a reagent that you can inject in the brain or in the heart, and you reach the same phenotypic level of information. And so basically, we started then at a certain point to ask ourselves if we could do more with these, more of these vectors, and basically the question is, can we develop a completely innovative system to use this vector for in vivo selection? Since they go very well on these organs, can we use them to select for new biotherapeutics? And the idea is this one. So suppose you have a library of factors and you want to check if one of these factors is beneficial after a degenerative condition. Suppose that in this library of factors which are cloned in plasmid, there is one which is good, which is this red one. Use this for a pooled production AV vectors. You will have a good AV vector, and then you inject them in vivo. Suppose you're interested in finding new biotherapeutics for myocardial infarction, you inject them in the heart. And each of these vectors will go to a different cardiomyocytes. Then you induce damage. In the case of the heart, you ligate the coronary artery. All the myocardial cells will die. But if a myocardial cell expresses a gene that is beneficial, then this will survive. It will have a beneficial, a selective advantage. You wait several weeks, and then you recover your vector from the heart. Each of the constructs has a barcode, so a degenerated 10 nucleotide sequence that distinguishes exactly that cDNA. And then you do next generation sequencing to sequence the barcode. And so basically, what you do then is simply compare the frequency of the barcodes here with the frequency of the barcodes here. And if some vector has been enriched here, it has a good probability of having been beneficial. Or you can take the same library and do different cycles of selection. So repeat the selection 1, 2, 3, 4 times. So we embarked in this project. Basically, this is like screening for neomycin-resistant in HeLa cells. You have neomycin-resistant genes, you mix with other genes which are irrelevant, you put the cells, you put the antibiotic, you select the clones. It is exactly the same, with the difference that the AV vector allow you to do this in vivo, which is completely impossible to do by any other means in these kind of non-replicating tissues. So Julia embarked in a project by constructing a library of factors that correspond to the mouse secretome. So we scan the mouse genome, check for the genes that contain a secretion signal, don't contain transmembrane portion. So these are secreted factors. And then she cloned the genes one by one. And then in another project, we obtained 800 different clones corresponding to microRNAs. The first screening that we did was for diabetes, and this is not finished, but I'm showing you the results because I think they are very exciting. So basically, we took the vector pools, we screened pools of 50 different factors at a time. We injected in the mouse, and then gave streptozotocin, and streptozotocin kills beta cells. Here, if a beta cell is transduced by a factor which gives a beneficial advantage, this is selected, and so Luca then recovered the beta cells, the islets, and checked for the selected genes. And what he found was that out of 1,100 genes that were tested, they all gave this kind of distribution. So basically, 1 means there is no selection. And you immediately see that there are genes that are selected out of the others. And when we check what the genes are by sequencing their barcodes, we discovered that For example, one of the protected genes is a prolactin. Prolactins are known to protect beta cells. Lactating mother with diabetes have a stop in progression of diabetes while they lactate. But all the others were completely unknown. So there are cDNAs, we really don't know what they do. They've never been described in the literature. Some of these are even from the RICAN collection, so they are just predicted cDNAs, not even the protein is known to exist. And even in these factors here, which were also enriched, there are several factors which we don't know, plus another prolactin, or interleukin-2, which is a strong inducer of Tregs. So we are very excited that here there is really the potential to find new biotherapeutics in the process of testing one by one these selected clones to see if they can constitute new biotherapeutics. Another screening that we did, which this is more visual, was to check, to test for factors that protect against ischemia. So we took mice, we injected both legs in mice with the library, and then we ligated the femoral artery on one leg only. And so basically we compared the frequency of vectors in the uninjected leg for the same animal with the injected leg without ischemia and the other injected leg with ischemia. And here we did the PCR with primers flanking the insert. So each of these bands is one or more of the clones we injected. You see we have a ladder. And basically these are the first round of screening. These are 2 mice from the first group of animals. This is the same mouse with ischemia, without ischemia. So basically you have to compare the ladder here with the ladder here and the ladder here. You see not much difference. We took all this material and recloned, did a second round of screening with AV vectors, and this time something comes out. For example, this band becomes a bit larger. We recloned everything, did a third round of screening, and this band now is really, really large. We take out these bands from the gel, sequenced, and with very excitement we found it was Ghrelin. I confess I didn't know what Ghrelin was at the time we did the screening, but you certainly know that Ghrelin was discovered in 1999 by Koima and collaborators in Japan. It is a 28 amino acid peptides that is produced by the stomach in condition of starvation. When an animal or ourselves are starving, we secrete this factor, which undergoes maturation in the stomach, and then it travels and it reaches a receptor which is present in the pituitary and convinces the pituitary to release growth hormone. At the same time, the receptor is spread in the brain. It convinces the brain, the animal, to adopt food searching behavior. It's the main reason why we search for food when we are under starvation. Why should it? We have selected Ghrelin was unclear, and so that was very interesting to us. We checked first if it works also in the heart, because we are interested to look if it works in the heart. This is an experiment in which we ligated the coronary artery in mice. This is an echocardiography experiment. This parameter is left ventricular ejection fraction, so it's the amount of blood that is pumped out by the heart at every heartbeat. Normally is around 70. After myocardial infarction, it goes down, and it progressively goes down, the white bars, after 2 months or 3 months after infarction. This is heart failure in mice, basically. The heart doesn't pump properly. If you give an AV vector expressing Ghrelin, then the function is preserved. This is another parameter, which is the capacity of contraction. And at the end of the experiment, this is the size of infarct, which is remarkably reduced. We discovered that this effect of Ghrelin is so strong because Ghrelin is a powerful inducer of autophagy. So we discovered that if we give Ghrelin to the skeletal muscle, to the heart, there is a strong transcriptional induction of several genes which are involved in the process of autophagy. Autophagy is a process by which organisms in the cell are self-eated by the cells in order to renew the cellular content, which is very clever if you think that a cardiomyocyte or a neuron are cells that are produced at birth and then they remain there for 80 years or up to 120 years. They are exactly the same cells. At least the cytoplasm has to be continuously renewed, and this is made through autophagy. And this is another marker of autophagy, is the inversion of the ratio between these two bands. So the translipidation of this protein is a marker of autophagy. And a particular form of autophagy that in the heart is very important is the autophagy of the mitochondrion, so mitophagy. And in fact, we discovered that if you give Ghrelin to the mitochondrion, then you see a lot of vacuoles, which are autophagic vacuoles because they express this LC3 protein in green, but contain a mitochondrion which is stained by mitotracher. So all these yellow dots are vacuoles of endocytosis, of autophagocytosis of mitochondria. And this is very important because after mitochondrial infarction, you have a strong burst in reactive oxygen species and strong damage to the mitochondrion. And if you get rid of this damaged mitochondria, then you do much well. And in fact, you increase the production of ATP, you decrease the production of ROS, and eventually you decrease the production of apoptotic cells. Here in red, you see tunnel-positive cells after myocardial infarction. There are a lot of cells that undergo apoptosis, but if you give Ghrelin, this number is very much reduced. Again, for the sake of time, I skip this part, but just to give you, I wanted to give you a flavor of this and just show you a table at the end of what we are doing for this in vivo library screenings, which we call the FunSel project, because it is fun, especially for me who go around telling this project, a bit less fun for those who do the experiments in the laboratory, but fun stays for functional, okay, selection. So we do this now, we're completing now selection directly in the heart after myocardial infarction. We have a collaboration with Stephen Hymans in Maastricht for viral myocarditis. We screen now here in the laboratory for cardiomyocyte proliferation and cardiac regeneration. This work in the pancreas is the collaboration with Fatima Bosch in Barcelona, Philippe Alban in Geneva. We started a project for amyotrophic lateral sclerosis with Cliff Svensson, who is the director of the regenerative medicine program at the Cedars-Sinai Hospital in Los Angeles. And we have a project here that's almost completed where we found cytokines that promote stem cell retention in the heart. Muscle ischemia gave rise to Ghrelin. We do have a collaboration with Tampere University for muscle regeneration after trauma. We would like to develop a gel that promotes regeneration of traumatized muscle to be sold to athletes to promote their recovery. And then we have a more serious project with Jeff Molkentin in Cincinnati for muscle dystrophy. The issue of how we can obtain cardiac regeneration is another one, and will be the subject of the presentation of Serena Zacchigna later, or tomorrow, I don't remember. And that's it, basically. A lot of people to be thanked, and so I won't name everybody, but it's just to say that all this is the core activity that we perform here, and thank you very much for your attention.